Welcome back to the deep dive. Our mission today is a little different. We're diving into pure strategy biotech strategy.
Mm.
We're not looking at a textbook and we're not analyzing some finished published study. We are cracking open the playbook, an investigational new drug submission, the IND. This is basically the blueprint for a revolutionary new antiviral package. Okay, let's unpack this. The therapy is called the viral eradication therapy, or VET system.
Mm.
And just looking at these docs, this seems less like a single drug and more, I don't know, like an integrated defense network.
It absolutely is. What we're seeing in this regulatory framework isn't just about killing a virus. It's about making the whole system resistance-proof. They call it a brutally optimized multimodal stack. I mean, it's designed to overcome the biggest weakness of our current antivirals, which is, mutation and escape. To do that, they've combined 6 absolutely crucial non-overlapping functions.
6 functions, all working at once. That sounds, well, exhausting for the virus.
That's the whole point. You've got a decoy blockade to stop it from getting in, then genome shredding to destroy its blueprints, pan-enzyme inhibition to paralyze its machinery,
Yeah.
and adaptive vaccination to build future immunity, real-time sensing so you know exactly what's happening, and this is critical reversibility, an emergency off switch.
So the mission for us today is to really figure out how modern biotech can piece together an escape-proof system like this,
Yeah.
moving way beyond the limits of, say, Paxlovid or monoclonal antibodies.
We're basically exploring the design principles of defense when you are absolutely determined not to lose the arms race ever again.
So let's start with that fundamental claim,
Right.
resistance proof. The IND protocol synopsis, it calls this therapy first in class because it's a dual decoy plus a genome editing system.
Sure.
That's a huge claim to make right out of the gate.
It is, and the regulatory justification has to be just as massive. They hammer this point home. The unmet need is how easily viruses develop resistance to single-mechanism drugs. Their primary goal is to drive the probability of viral escape mutations down to near 10 to the minus 8.
Okay, I hear 10 to the minus 8, but what does that number actually mean for you listening?
It means that with current drugs, you might see resistance in 1 out of every, say, 100 or 1,000 treatments. A probability of 10 to the minus 8 means you would statistically need to treat 100 million people for just one resistant virus to maybe emerge. You basically close the exit door before the virus even knows it's in a fight.
That is incredibly aggressive.
Yeah.
And strategically, they're highlighting 4 major claims here. They're not just selling a pill.
No, not at all. They're selling a new way to handle a pandemic-level threat. So first, like we said, is that it's resistance-proof. Second, and this is huge for regulators, it's self-terminating. It has a suicide switch.
A suicide switch. We'll definitely come back to that.
Third, they've built in real-time diagnostic integration, a patch you wear.
And the 4th point, this is the one that really tackles global access, right?
Exactly. Cost effectiveness. The projected cost is around $25 per course. That puts it way below most current treatments.
So they're solving for efficacy, safety, diagnostics, and global supply chain, all at the design level.
Yep. And to test it, the Phase 12A clinical trial is lean and fast. 12 healthy volunteers, 24 mild COVID patients, and a dosing schedule on day 0 and day 3. They want data, and they want it fast.
Okay, here's where it gets really interesting. Let's dig into the components, the actual attack. We'll start with the decoy. The SARNA decoy, ACE2 FCABE bispecific. I'm already swimming in acronyms, so let's break that down. What's a decoy, and why is this one bispecific?
Well, think of the virus as a key. Its RBD, the receptor binding domain, is the key, and the ACE2 receptor on our cells is the lock. A decoy is just a fake lock, a really good fake lock that grabs the virus before it can find the real one.
Got it.
Now, this one is bispecific because it's like a molecular bear trap. It doesn't just block the RBD key, it simultaneously blocks a second crucial spot on the virus called the HR1 groove. That's what the virus uses to actually fuse with our cell membrane.
So it's stopping both entry and fusion.
At the exact same time. It makes resistance exponentially harder.
And the delivery system, SARNA, that's key here too, isn't it? It's not your standard mRNA.
Great. SARNA stands for self-amplifying RNA. Think of it like a blueprint that also contains its own little photocopier. Once it's in your cells, it makes way more therapeutic protein than normal mRNA, so you need a much smaller dose.
And what about the stability they're claiming? 3 to 5 micrograms per mil in the serum for 10 days. What does that feel like for a patient?
That's the real-world impact. High stability for 10 days means you get long-lasting coverage from just 1 or 2 doses. No daily pills, no infusions. You just have this constant therapeutic shield in your blood for almost 2 weeks.
So the decoy stalls them at the gate. But what if a virus slips past and starts to replicate? That's where the genome shredding comes in, right? The Cas13 chop lock system.
That's your second line of defense. Exactly. Cas13 is a type of CRISPR system, but instead of editing DNA, it's armed to find, cleave, and just destroy viral RNA, the virus's operating manual.
But the Cas system, that's where regulators get nervous. How do they make sure the virus can't just mutate and evade the Cas guides?
They use a combination of brute force and really clever targeting. The system uses 200 hyperconserved 20mers as guides. So they're not targeting random bits of the virus. They're targeting 200 tiny sequences that are absolutely essential for the virus to function, sequences that have barely changed across all known variants.
It's like taking out the engine instead of trying to slash the tires.
Exactly. If the virus mutates these sites, it can't replicate anyway. It's a dead end for them.
Okay, which brings us to the critical safety question. If you're injecting what is essentially a gene editing tool, even if it's for RNA, how do you convince regulators this isn't some risky permanent gene therapy?
This is, I think, the most brilliant strategic move in the whole submission, the miR-122 auto-off switch. So miR-122 is a microRNA that's naturally abundant in the liver. They engineered the Cas13 mRNA with a little sequence that miR-122 recognizes. When the mRNA hits the liver and sees that miR-122, it triggers its own self-destruct.
A literal self-destruct sequence.
It is. And the effect on persistence is just dramatic. It drops the half-life of the Cas13 mRNA from a potential 14 hours all the way down to just 45 minutes.
45 minutes.
Right. And that lets them argue to the regulators that this therapy is purely transient. It's not a living drug. That just massively lowers the regulatory risk profile. It's the strategic lever that makes the whole thing possible.
Okay, so we've got the decoy and the shredder, but the VET system also includes agents that inhibit viral function and then boost long-term immunity. Let's talk about this bispecific FN3 monobody.
So the monobody is the paralyzer. An FN3 monobody is a synthetic antibody fragment. It's very small, very robust, and its job is pan-enzyme inhibition. It's not variant-specific.
And what are the two big enzymes it's hitting at the same time?
It's hitting the core of the virus's replication machinery. First, RDRP, the polymerase that copies the genome. And second, 3CL-Pro, the main protease that cuts up proteins to assemble new viruses. You inhibit both replication and assembly, and the whole factory just grinds to a halt. This dual targeting gives it incredible potency. We're talking an IC50 of 1 nanomolar, which is 100 times better than single-site inhibitors.
And the manufacturing is also a huge differentiator here, right? They're moving away from the old ways of doing things.
Precisely. They're using cell-free protein synthesis, or CFPS. So instead of growing these massive vats of cells in bioreactors, they're basically making the protein in a test tube. It's incredibly scalable, it's fast, and it's cheaper than traditional methods.
And it avoids a lot of supply chain risk.
A ton of it. And look at the formulation. They're using 15-kilodal polysarcosine instead of PEG, which is common in lipid nanoparticles. A lot of people develop an immune response to PEG anti-PEG IgM, which can make drugs less effective over time. By using polysarcosine, they strategically design out a known cause of treatment failure.
That is really thinking several steps ahead. Now for the long-term defense, the 64-peptide hydrogel vaccine, the fourth function.
This is the immune system's training program. They used advanced bioinformatics to identify 64 specific protein fragments, peptides, that give you extremely broad coverage against variants. They're guaranteeing 92% VOC coverage.
And how do they deliver those peptides to make sure you get a really strong, sustained immune response?
The delivery is very strategic. They use a PLGA hydrogel. It's a biodegradable scaffold that acts like a slow-release depot, so the peptides are shown to the immune system over time. And it uses a fascinating adjuvant, a booster, called TKK301, which is actually a melatonin analog. The target is rapid mucosal immunity. Specifically, they're aiming for sterilizing IgA titers of at least 1 to 256 by day 7 after the boost.
Sterilizing IgA. That means immune defense is right where the virus gets in the nose, the lungs. So to sum up the core therapy, you have an immediate attack with the decoy and monobody, rapid termination with the Cas13, and a long-term defense launch with the vaccine.
It's a comprehensive offense and defense package, all delivered at the same time.
The genius here is that they didn't just design a cure, they built a full-service platform. They've integrated diagnosis and safety in a way you just don't normally see in a drug IND. Let's get to those final two functions. First, the real-time diagnosis, the viral nickase sensor.
This attacks the biggest logistical flaw of most antivirals. You have to give them early to work, but diagnosis is often slow. This sensor is a CE-marked microneedle patch that you wear for 5 days, and it detects the virus.
So what makes this a game changer over, like, a standard rapid antigen test?
Time and accuracy. They're aiming for over 95% sensitivity compared to the gold standard qPCR test, with results in only 15 minutes. If you can confirm an infection in 15 minutes at home, the window for effective treatment with this VET system opens immediately. It removes the biggest bottleneck.
And then the ultimate regulatory reassurance, the emergency stop, the literal universal kill switch.
That's right. The sixth function, reversibility. This is the DGCR8 SIRNA mechanism. If a severe adverse event happens, this is the emergency parachute. It's delivered intravenously, and the SIRNA targets an enzyme, DGCR8, which is essential for processing microRNAs.
So by knocking down DDCR8, what happens to all the therapy's components? The SARNA decoy, the Cas13?
They can't be processed properly, and they get cleared from the system incredibly fast. The documentation promises to trigger 90% DROSHA inhibition within 30 minutes, and it ensures all the mRNA components are cleared below the limit of detection by day 10. That level of controlled reversibility is a massive selling point to regulators.
And on safety, let's just quickly revisit the toxicology findings for everyone listening. What were the key non-clinical results that addressed the big worries?
They went right at the two biggest fears. For the Cas13 genome shredder, they confirmed no off-target edits with guide-seq validation. It only hit the viral RNA, not human RNA. And for the decoy system, they found no liver tropism, so it didn't build up in the liver, and no anti-drug antibodies, or ADA, in the primate models. The system is just engineered for maximum effect with minimal side activity.
And finally, let's talk scale and cost. The big picture for global impact. They are targeting a 10 to the 9 dose production capability. 1 billion doses.
It's immense, and it's only possible because of that modular design we talked about. Using CFTS for the monobody and modular GMP suites for the nucleic acids, they think they can keep costs down while scaling up fast. The cost breakdown they provide shows exactly how they get to that $25 goal. The decoy is about $8, the monobody is $6, the sensor is $4, and the rest is formulation and packaging. This is a system designed for global distribution from day one, not just for wealthy nations.
So this VET system, as it's laid out in this IND filing, is really a showcase of modern, aggressive biotechnology. It takes the 6 functions we've discussed and pulls them together into a system that is fundamentally resistance-proof, self-terminating, and highly cost-effective.
It really reflects the lessons we've learned, that infectious disease defense has to be multi-pronged and it has to be scalable. And the aggressive timeline just confirms the priority. R&D submission targeted for Q4 2024 with first-in-human plan for Q1 2025. It truly is, as they say in the source material, a viral eradication system, not just a treatment.
Which leaves us with a final provocative thought for you to chew on. If future therapies start to integrate diagnoses like the sensor, multiple targeted attacks like the decoy and Cas13, and immediate safety fail-safes like the kill switch, does this holistic platform approach fundamentally change how we should view drug development for infectious diseases? What does ready to make some viruses regret existing actually look like in practice?